It's an absolute pleasure and honor to have Matt Jones give us a talk. He just recently, in September, started as an assistant professor at MIT, both in the Department of Biology, the Koch Institute, and IIMS. Previously, he did his PhD in near USF's work lab in UCSF. Near is a long-term and dear collaborator of mine, so it's funny how those worlds cross. And did some really beautiful, some of the earliest lineage tracing work that published really well. He then went on to do his postdoc in Stanford University with Howard Chang to work on, amongst other things, ecDNA. And I think that's the topic of his talk today. So, delay it no longer. So, Matt, thank you so much for joining. I know there's an interest in the IICD community around ecDNA. So, excited to hear you talk.
Wonderful. Thanks so much, Yeller. And I mean, truly the honor is all mine. I think this probably started going remote, like probably during the pandemic, probably these meetings. And I remember during the pandemic watching almost all of them. So it's like, it's real full circle and a real honor for me to actually be giving one of these presentations. So thanks so much for the invitation. So yeah, right on point. Today, I'll be telling you about some of the work that I did during my postdoc, but also some work that was just published last fall and even some unpublished work. Really looking at how we can quantitatively profile and model extrachromosomal DNA amplifications in cancer. And so, as many people will appreciate, tumors are very complex and dynamic systems composed of many different types of cells. They're governed by evolutionary processes, and I like to say from the moment that a tumor is born, it's evolving. The genetic landscape of a tumor is changing as cells uncontrollably proliferate, the epigenetic landscape, the metabolic landscape, and even the structure of the tumor is dynamically changing. And understanding the different dynamics of governing the system and the therapeutic opportunities is really difficult because oftentimes all we have access to is a single snapshot of the tumor and all the different complexities from that single snapshot. So if we're trying to make predictions about how a tumor will develop into the future, maybe in the presence or absence of targeted therapy, this is a very difficult and obviously clinically relevant challenge. And the central thesis of my group is that probably the best piece of information that we have is understanding the history of the tumor, how the tumor got to where it is today, and how different types of evolutionary pressures are affecting what we're able to observe, and hopefully we're able to extrapolate the future. So this is the central goal of our lab's research, is to understand the evolutionary principles that give rise to cancer, ultimately not just to identify new therapeutic targets to control the tumor, but also hopefully build predictive models that allow us to forecast further into the future how the tumor will respond or continue to develop. And so the unifying theme of our group is we're an interdisciplinary hybrid group that uses both computation and new technologies to study these processes. And on one hand, we are able to measure time with a set of evolutionary modeling approaches, including lineage tracing technologies that, of course, Deanne Yang has pioneered, sitting in the audience, and I've had the pleasure and privilege of working with it closely. So we can use these kind of lineage tracing technologies as well as evolutionary modeling techniques to measure and model time. And then we also make use of a lot of these new genomic technologies that allow us to make really high resolution measurements of the molecular state of cells.
Yeah, I hate to interrupt, but the right side of your slides are not showing up on Zoom at all. I don't know if that can be fixed easily or not. Sorry.
Is that true? I'm looking at the...
So what happens is after the word for this title, it says computation and everything to the right of the word and is not showing up.
All right, let's see if we can just reshare the screen. And if anyone has any idea on how to fix this, I'm all ears. Is that any better?
Now swap, do swap. Yep, that's perfect.
Okay, but now.
Maybe you can do the book view instead of the slide view.
So let's see, I just want to. want to stop. I want to start mirroring stuff.
Can you try something?
Yeah.
You go to the presentations.
Yeah.
And instead of slide view, you click the V to the left of that little forward.
Oh, I'm gonna share this.
Click the little this up here now. And see if you can maximize it here and see if it works for that. Let's do that.
All right, now on Zoom, does that look better?
Yeah.
Okay, great. So we can measure evolution, we can measure time with these evolutionary approaches, and then we could also use new single cell and spatial technology to get really specific molecular profiles of individual cells in their spatial context. And linking these two types of technologies and techniques together, we're hoping to build predictive and mechanistic models that allow us to recapitulate the evolutionary pressures that can give rise to the types of patterns we observe in space and time. And so a major, our lab is invested in both developing these technologies as well as using them. And the focus of my talk today is talking about how we're using these to understand, I think, unique aspects of how the tumor genome is evolving. And that's specifically with respect to focal amplifications. And so I know many of you are probably aware of how these focal amplifications work. Basically, copy number changes are very common in cancer. You often hear about MYC amplified or EGFR amplified cancers. And dating back to the 1960s, people started to appreciate how these amplifications could happen. The first sort of amplification could happen on the chromosome. And so these would form large regions that were stained with the same FISH probe, and they would form homogeneously staining regions called HSRs. And then the second form of amplifications, originally thought to be very rare, maybe 1% of cancers, were extrachromosomal, where it appeared that a region of the chromosome had been excised off, circularized, and formed as a separate pool of circular DNA elements within the nucleus of cancer cells. And like I said, these were rare occurrences, but both types of amplifications were appreciated to carry oncogenes, regulatory elements, and could appear at very high copy numbers as well. And for about 50 years or so, there was a great line of work investigating the biology of these ecDNA, particularly amplifications. But really our appreciation for the importance of these took off in the early 2010s when we started doing deep whole genome sequencing of large patient cohorts and identifying these ecDNA. And the first lesson that came out was that actually ecDNA were actually much more prevalent than originally appreciated. So instead of 1% of cancers roughly, it was almost a quarter of cancers that were analyzed at diagnosis. So that's roughly the number of patients who are diagnosed with a KRAS driver mutation, for example, and you can appreciate maybe the scale of how many patients that impacts. And even more importantly, we found that ecDNA was increased in prevalence in aggressive cancers like glioblastomas, lung cancers, breast cancers, and later stage cancers, and in drug-resistant and metastatic cancers as well. And when we started profiling even more patients, this is from a recent study in the Genomics England study, about 16,000 patients were considered here. There was a remarkable significant difference between patient survival of those patients driven by ecDNA-driven tumors versus by tumors that were driven by chromosomal amplifications alone. And I like to say that the focus on our research program, a lot of it is understanding this gap between what's driving the increased mortality and aggressiveness of these ecDNA-driven tumors. And so we started to understand what makes ecDNA unique, how they're affecting the phenotype of individual cancer cells. And I'll tell you just a little bit of what we started to appreciate. The first thing is that ecDNA promote a very permissive epigenetic state. And so in about 2018, there was a study that used an optical assay to measure chromatin accessibility, and we were able to partition out the chromatin accessibility coming from ecDNA versus from chromosomal regions, and we found that ecDNA was actually much more accessible than the relevant chromosomal regions. So the same oncogene or the same genomic context on ecDNA more accessible. Using nanopore sequencing and an assay we called CRISPR-Catch, our team also found that accompanying these higher accessibility of ecDNA were focal demethylation events on the ecDNA specifically. And this is particularly relative to an oncogene EGFR amplified, but if you look at the promoter sequence around the chromosomal EGFR locus, it was hypermethylated compared to the ecDNA. And so this ultimately leads to, on a per molecule basis, higher rates of transcription per copy number. So not only are ecDNA extremely high copy numbers, but they're also more productive. The second rule, or the second thing that ecDNA do that I personally found very interesting from a tumor evolution perspective is that they actually violate how we think that genetics propagate within a population. So we probably have a mental model of how tumors evolve, where mutation happens and can pass on to daughter cells, and higher fitness mutations take off. ecDNA violate this rule. And the reason they do this is because they lack centromeres. You can see that they don't, the amplification here shown in the little dots red, which are the ecDNA, they don't have centromeres. And so every single cell division, individual molecules of ecDNA are shuffled up and passed on randomly to daughter cells. And so you can imagine this is going to lead to rapid increases in intratumoral heterogeneity and even drug resistance, because if you're targeting something on that ecDNA, quickly you can generate a daughter cell lineages that lack that ecDNA molecule at all. And so they're violating the rules of tumor evolution in some of the worst ways. And our understanding of how they're doing that is still underdeveloped. And so a reason why our understanding is underdeveloped here, I think, is that our existing approaches to study ecDNA often lack resolution and functional insight in how individual ecDNA amplifications are affecting the molecular state of the cell and the tumor. So let me give you a little bit of history. How do we historically measure ecDNA content? The first one is with fluorescence in situ hybridization, or FISH-based approaches, where you can design a probe that can target a region of the genome and it'll light up with a cytogenetic assay. Shown here are a special version of these FISH assays, which are metaphase FISH assays, where you can partition out which particles of the DNA is on ecDNA versus on the chromosome. And these little dots that we're highlighting here are ecDNA, and the other blue dappled regions are chromosomal DNA. So we can get a measurement of if a tumor has ecDNA or not, but it's really low throughput. We need to look at one cell at a time, and they're very large probes. So we actually don't have that much even genomic resolution. And of course, there's no phenotypic information that accompanies this. The second set of approaches that really actually drove the field forward about 15 years ago was the advent of techniques that could use bulk whole genome sequencing to look at ecDNA. And shown right here is what we call a breakpoint graph that summarizes all the rearrangements internal to an ecDNA, so we can decompose this into cycles that actually are the circular DNA particles. And so even though we have high resolution genomically, the bulk readout is actually hiding the single cell nature, so how frequent ecDNA appear within a tumor, as well as any kind of phenotypic information as well that's accompanying the presence or absence of ecDNA within a cancerous cell. And so these limitations, I think, really are limiting our ability to understand the subclonal resolution of ecDNA and the behavior of ecDNA within cancerous cells. And so for the rest of my talk today, I'm going to tell you 3 stories. 2 that have been previously published that use new techniques to look at the segregation and behavior of ecDNA within cancer cells, and some new findings we have from this. And then I'll end with some new methodology that we just submitted that we think will continue we'll be able to continue building on. We hope also serves to motivate the field as well. So the 2 stories that I'll tell you first are largely collaborative studies that I led during my postdoc with colleagues in the lab. One using new techniques for imaging-based assays to look at really high precision, the genomic context where ecDNA are appearing within cancer cells. And the second one is using single cell assays to measure the subclonal heterogeneity, as well as the segregation dynamics across cells and experimental systems. So the first of these, how ecDNA, how we can use spatial or imaging-based assays for these. So as I said, ecDNA have a very unique epigenetic landscape. They're highly rearranged as well. And so the way this story started out is actually just from a pure curiosity perspective. We're going to run Hi-C on ecDNA positive cells, which capture the chromatin conformation of the genome. And we're going to ask if ecDNA are occupying specific conformations at the chromatin level. So we took one of our cancer cell lines with ecDNA, it's COLA 320, it's a colorectal cancer cell line. And when we ran Hi-C on it and started looking at the data, what we're struck to find is look at the Hi-C maps and taking the viewpoint of our individual ecDNA cancers, our individual ecDNA molecules, we found is that there were contacts that the entire ecDNA molecule targeted against very specific regions of the chromosome. We called these pretty unimaginatively ecDNA interacting elements. There are about 68 of these we found in this one cancer cell line. And we thought of many different models of how this could possibly occur, some biological some not very interesting. It turns out these are artifacts from structural variation that appears on the ECDNA that we previously didn't actually appreciate from all of our sequencing efforts. And so these are the by and large, what these 68 elements are insertions of different parts of the genome onto the ECDNA that are carried along. And when we started profiling what these artifacts were, these structural variants, what we found is that they're actually enriched for repetitive elements. So these repetitive elements, or transposable elements specifically, these are the residue of largely viral integrations that we have carried along over our course of evolving. There's many different types of transposable elements, and we were starting to see that a wide variety were contained within these 68 ECDNA interacting elements. And specifically, we noticed that there were a large fraction of these line 1 elements, which are the only autonomous or only elements that are capable of autonomous vector transposition within their human genomes. And specifically, we started focusing on this one element, EIE14. And so we ran ultra long read sequencing. So we basically nanopore sequencing, we could get reads up to megabase or larger in scale. And we reconstructed the amplicon for this ECDNA in this colorectal cancer cell line. And we found that that there was a strong evidence for this one element, EIE14, to transpose onto this EIE into this location just upstream of MYC, which is a common oncogene that's carried on this ECDNA. And so we were initially excited that, maybe we're capturing the somatic retrotransposition event. As it turns out, this element is not capable of somatic retrotransposition. It's an ancient element, it doesn't have the kind of enzymatic ability to do this. So we think this is actually a passenger element that probably existed in a fraction of individuals. It probably pre-existed as kind of a germline event that got amplified as a passenger event. But then the story becomes perhaps even more interesting when we start to look at the behavior of this element in the ECDNA context. And so this is where we start using very quantitative imaging-based assays. So specifically, we use an assay called ORGA, or optical reconstruction of chromatin architecture. We can design FISH probes essentially that target very small regions of the genome, roughly a kilobase in scale. And we can look at the localization of that FISH signal intracellularly with respect to other types of elements. Specifically, we're looking at MYC, this oncogene I just told you about that's amplified here, as well as PBT1, which is a non-coding RNA that also exists within the same chromosome 8 locus. And shown right down here on the bottom left-hand side, this is generally what this circle looks like of the amplicon, the circular amplicon, where you might have multiple EIE14s amplified on this ECDNA, multiple MYCs, and multiple PBT1s, but all in the same circle. And so because we're able to quantify subcellularly within the nucleus how the spatial organization of these elements are, we can actually look at the spatial distribution and look at how these different spatial distributions might indicate the behavior of these elements. And what we're really struck to see is that in really small regions, at really close distances, there is increased clustering between the MYC oncogene and EIE14, even more than this other PBT1 element, which has actually been validated as actually enhancing MYC expression as well. And so what we're surprised to see was that this passenger line elements seem to be clustering according to what would be perceived to be kind of an enhancer-like behavior. And so the next assay we did was we put this element onto a luciferase assay where we could measure the promotion of expression of two different luciferases, nanoluciferase and powerful luciferase, with respect to different kind of enhancer elements. Like I said, we used PBT1 enhancer as a positive control. And we found that actually this EIE14 actually had quite significant ability to enhance the expression of both these different types of luciferases. And so this really was surprising to us that this inert line element that appeared to be co-amplified with ECDNA just randomly could actually gain this ability as an enhancer. Now the question is this also behaving this way in the chromosomal context, or is it just in the ECDNA context? And as you might imagine from all the things I've said so far, there's remarkable differences between the ECDNA and chromosomal context. What we found is that at this enhancer locus, or at this EIE14 locus in the COLO320 DM context, that this locus actually had several indications of enhancer function. This HK27 acetylation mark, which marks active enhancers. And if we looked at several other cell lines, we could find that this region was actually marked by repressive histone marks, specifically H3K9 trimethylation, which is actually often deposited at translocal elements to silence them. And even more strikingly, when we looked in the isogenic cell line pair that doesn't have this ecDNA, but rather this ecDNA has integrated into the chromosome, what we found is that this EIE14 was expressed lowly, but expressed in the DM context, but not expressed at all in the HSR context, which really does seem to suggest that something about this ecDNA context is responsible for reorganizing the genome into a permissive epigenetic states. And I'll say before jumping to the next story that we've identified and molecular characterized how these ecDNA molecules are hijacking transposal elements as enhancers. But really, there's other groups that have started to profile patient cohorts in bladder cancers, as well as metastatic castration-resistant prostate cancer, and also found that these transposal elements are enriched on ecDNA, where they might be driving viral mimicry pathways, new transition factor binding, for example, with androgen receptor binding, as well as potentially even new ecDNA amplifications. And so this serves as kind of an example of how we're using new imaging type approaches to get really high resolution pictures of the organization within the nucleus of how chromatin is conformed around ecDNA versus chromosomes. So with that, I'll jump back into genomics a little bit and talk about how we're using genomic approaches, single-cell genomic approaches, to study the segregation dynamics of ecDNA. And so let's zoom out really quick and think about the how ecDNA are organized again in the cell. So as I said before, ecDNA are heterogeneous. They're really high copy numbers. And as a corollary of that, what we've observed is that distinct versions of ecDNA can coexist within cells where they can actually activate one another synergistically. And shown right here is kind of a schematic model of this. And the way we think about these ecDNA hubs within the nucleus of the cell is that they're largely driven by enhancer promoter contacts. And our group has shown previously that if you break up these hubs, you can stop transcription, actually cells start to die. So they're actually dependent on these intermolecular interactions between ecDNA. In an analysis of TCGA, a large cancer genome study, what we found is that of the fraction of tumors that have ecDNA, about 25% of these have indication that multiple ecDNA species exist that are capable of interacting with one another and synergistically driving gene expression. And so the synergistic expression between distinct molecules of ecDNA is quite interesting, but it actually runs counter to what I told you earlier in the beginning of my talk, which is that ecDNA are violating the rules of evolution and their random segregation. And so this is a distinct tension. If ecDNA are capable of randomly distributing amongst between cells during cell division and breaking up the combinations, how are these synergistic interactions being preserved? And so this is a major question that we posed early on in my postdoc, was how these cooperative interactions are being preserved and maintained despite the random segregation. And this really requires being able to look at amplifications at single cell resolution, understanding the distribution and temporal dynamics over time. So when we started profiling individual cells, we went back to this metaphase spread assay. So this is a single cell that we're looking at here. The blue dots here are DAPI-stained chromosomes, and then the red and green dots are distinct amplifications on ecDNA, MYC, and FGFR2. And what we were struck to find first was that the copy numbers of these two distinct ecDNA foci were really high and co-occurred in almost every single cell that we observed. So there was something about these cells that allowed them to maintain these synergistic interactions over time. We then applied single cell attack sequencing, and we could get copy number estimations from single cell attack sequencing. And what we observed that not only in this one particular cell line were the ecDNA regions co-amplified, but that there was a remarkable co-segregation or correlation signal. And this is really surprising because when we first did this assay, we'd expect that, okay, even if these are co-occurring within cells, there's no way that the correlation between copy numbers would be there because of the random segregation that we and others had described previously. So we found this kind of correlation result. We knew that there was something else driving this kind of behavior. It was even more striking to see that we found this across 6 different cell lines, across multiple oncogene pairs, 3 different orthogonal methods using metaphase, single cell attack, and then also this assay called Circle-seq, where we can sequence the actual DNA at single cell level. And so, motivated by this, we started thinking about how exactly this could come about. And there's two different ways this could happen with reason. The first way is that maybe there's this previously undescribed way for these synergistic combinations of ecDNA to be packaged together during cell division and passed on to daughter cells. And we call this co-segregation. And then the second way this can happen, I guess you would all imagine, is just obviously maybe this selection is just so strong that over time you would just shape the copy number distribution, the joint distribution, just from selection alone. And so we started off just showing or seeing if we could detect this co-segregation mechanism. And the way we did this is we started with an imaging assay where basically we could press pause during cell division. So cells that were directly butting off one another, daughter cells, and we tried to measure the ratio of these two different ecDNA foci, the signal between the daughter cells. And we hypothesized that if we saw correlation in the daughter cells, that would be indication within a single cell division, you could generate this correlation, and that would be direct evidence for co-segregation. And shown right here is an image of what this looks like. And the SMooth16 cell line I've been telling you about before, what we found is that, yes, indeed, in a single cell division, you could generate this type of correlation between the two ecDNA species. So there was something that was allowing these interacting species to co-segregate together over time. Now, we also saw this across 4 other cell lines, multiple oncogene pairs, multiple different types of cancers, varying amounts of this co-segregation that we measured. So some had a really high co-segregation, some were around 0.3 kind of correlation. So varying amounts. And we thought how this could possibly be happening. And we were very lucky to just hypothesize that maybe it was these intermolecular hubs themselves that could drive the co-segregation. And if we broke up the hubs, maybe we could actually inhibit the co-segregation itself. And so that's what we did, is we broke up the hubs by actually stopping transcription with a compound called triptolide, and we saw that we could get significant reduction across all the different cell lines by stopping these hubs. And so it seems like these two different conflicting kind of observations, the synergy and the random segregation, are actually related to one another. Then if you break the synergy, the synergistic interactions, you can actually restore the random segregation. So in a way, these cancer cells are kind of getting the best of both worlds. Now, going back to co-selection, it was totally reasonable that co-selection could also be driving this type of behavior, but to what extent were these complementary or redundant sort of forces? And this is a really difficult question to ask. It's difficult to ask questions experimentally about selection in this kind of context. And so the tool that we decided to go to was evolutionary modeling. And this was actually a lot of fun because when I was an undergraduate, my first research experience was in an evolutionary modeling lab when we were looking at the evolution of humans. And now I got to actually use those kind of tools to actually model in forward time how cancer cells were dividing. So we set up our model in such a way that we could parameterize a founder cell with different ecDNA amplifications. We decided on 2 different amplifications that you could put in many different types of amplifications, species, at different copy numbers initially. And then every single cell division, we could specify the degree of co-segregation we'd expect. And so basically, if you have a co-segregation of 1, that would mean that every single species would perfectly co-segregate with the other species. And if co-segregation coefficient of 0 would mean that you have perfect random segregation. And then you can also specify selection as well. So an expectation, how much faster will a cell divide given its content of ecDNA species at what copy number. And we could generate very large cellular phylogenies, and we did millions of these forward time simulations where we titrated these parameters in relation to one another and studied how these different parameters left distinct signatures on the copy number distributions that we ended up observing. And just summarizing a lot of work here that I won't be showing, that I'm happy to talk about, is what we found is actually these 2 different parameters were actually partially, they were complementary and partially orthogonal. So shown right here is a kind of schematic of how we thought about it. So imagine you have a full space of all possible joint distributions. What we found is that the co-selection selected for cells that were co-amplifying these ecDNA, but couldn't actually act quick enough to place any kind of stoichiometric balance between the ecDNA copy numbers themselves. So it wouldn't induce correlations between the copy numbers. Whereas the co-segregation coefficient that we put in was actually capable of kind of squeezing this distribution, shaping it into kind of what we actually observed experimentally. And so because of these are, complementary, partially orthogonal parameters that are affecting the joint distribution, we actually started to estimate these on real samples, including tumor samples in our experimental samples as well, using ABC or approximate Bayesian computational methods, which I think many of you in this institute are using as well to kind of fit these parameters to experimental observations. And so this is a very powerful tool that showed us that co-selection and co-segregation were actually operating together to shape the copy number distributions in patient samples as well. Now, the real power of this is going also forward in time. So we learned the evolutionary rule shaping the distribution. Can we make predictions about the future? And cancer, oftentimes what we care about is the future with respect to a drug perturbation, let's say, whether a tumor respond to the drug or not. And so we set up a very simple assay. We took the same cell line that I've been showing you all along the way. It's SNU16. It has two different oncogenes amplified on distinct ecDNA, like I said, FGFR2 and MET. And we asked a simple question. Because these cells are co-segregating with their ecDNA, what happens to the species if you target only one of them? And so we targeted FGFR2 with pemigatinib. It's a clinical grade inhibitor of FGFR2 or FGFR in general. And we asked how the copy number dynamics is going to change for FGFR2 ecDNA, but also MYC. And could we actually bring down the whole house by just targeting one? And so we calibrated our expectations by first doing a lot of modeling. And so we went back to this evolutionary modeling setup, and we started simulating tumor populations in the presence of co-segregation. And what our modeling suggested was that if you have co-segregation, both copy number trajectories will actually be kind of responding at the same time to pemigatinib inhibition of FGFR2. So you can get kind of like this oscillation that's really responsive to the pemigatinib treatment schedule. Whereas if you didn't have co-segregation, if things were just randomly segregating, you would have quickly decoupled the trajectory of copy numbers from between MYC and FGFR2. So FGFR2 would respond to pemigatinib, but pretty soon you'd develop a lineage that only had MYC amplification, and that copy number trajectory would take off because that lineage would be resistant. So our modeling seemed to suggest that we can interpret the behavior of these parameters with respect to the copy number trajectories over time of these two different drugs. And so we hypothesized that if you had co-segregation, you could actually get indirect targeting of MYC, even if you're only targeting FGFR2. And I think some really cool stuff happened. We actually did this experiment in vitro. And so when we started doing this experiment, once again, the SNU16 cancer cell line, what we found is over the first 2 weeks of experiment, actually our predictions played out that we could get direct reduction in the copy number trajectory of both FGFR2 and MYC. However, if we continue treating over a full 6-week drug time course, we found is that while FGFR2 copy number plummeted, the MYC copy number started to take off. So we were a little disappointed, but of course, this is kind of what you'd expect from the resistance. Like maybe it'll work at first, but eventually you're going to generate a lineage, even if you have co-segregation, because co-segregation is not perfect. You have a loose correlation. But it becomes even more interesting when we introduce the drug holiday in the middle of the experiment. And when we have a drug holiday where we stop treating with pemigatinib for about 2 weeks and we put it back on for 2 weeks, what we found is that actually we could restore this oscillating behavior. So we could restore sensitivity with the presence of the drug holiday. And so to start to get the mechanism of how this actually happened, we went back to the metaphase spread assay, which gives us a lot of information about the localization of these ecDNA. And what we found is that in the DMSO condition, so the control condition after 42 days, every single cell had a good portion of ecDNA carrying both FGFR2 and MYC on separate circles. In the pemigatinib condition, the MYC foci had taken off, but all the FGFR2 foci had integrated into the chromosome. And so this is really remarkable because this is not the kind of resistance mechanism that we thought we were going to identify, but rather this is completely separate. And realistically, what this means is that resistance is emerging by breaking the co-segregation mechanism altogether. Because once MYC is amplified as ecDNA and all the FGFR2s on the chromosome, you restore random segregation from the viewpoint of FGFR2. And you can just select for cells with high and higher MYC. There's a lot of questions here as to why this integration is actually advantageous. I'm happy to talk about my thoughts, but this is an active area of our research. And so summarizing this is that the drug resistance actually, in this observation of co-segregation, actually generates, I think, new insights into how drug resistance is happening. And it's not that the cells are entering into a new, molecular state per se, but the organization of the genome, ECNA, as either, or the amplifications out of ECNA or on the chromosome actually has direct relevance as to the responsiveness of these cells to pharmacological inhibitions. And so, and then the good news is also for our modeling, it wasn't that our modeling was wrong, it's just that we hadn't taken into account that there's actually a state switch between how these amplifications were evolving. And so I think that this is another thing we can start baking in order to understand mechanistically how these cancer cells are evolving. And so as I'm starting the group, I'm continuing to be interested in how kind of new techniques are able to inform on new areas of biology, especially in ECNA right now. And I think some of the major challenges is continue to try and quantitatively profile and study ECNA, both in vivo, but then also in the context of these kind of drug resistance assays as well. And as I kind of mentioned before, some of these challenges include we don't really have great ways of measuring in high throughput the frequency of ECNA positive cancer cells within a population. Oftentimes, if we take a tumor from a patient, we profile it with whole genome sequencing, which really masks a lot of the subclonal single cell heterogeneity. We don't know about the evolutionary timing of when ECNA emerge, how often they emerge, and what the subclonal architecture is of ECNA origination. And going back to this drug resistance case, we also don't know how the context of the amplification changes, how often, what are the kinetics of it as well. And lastly, we don't have any good methods right now of actually tying the molecular state of a single cell that we observe or its spatial context to its amplification landscape. And many of these genes that we see amplified on ECNA are common oncogenes like MYC and EGFR, but we also see transcription factors. We also see immunomodulatory elements as well, which really does seem to indicate that there's something molecularly beyond just making the cell grow faster that these ECNA are amplifying. And so one of the things we've been trying to do is develop new techniques that allow us to detect ECNA de novo from single cell assays. And the hypothesis we had was that going from the random segregation of ECNA molecules themselves, we thought that basically ECNA amplifications would have a much wider distribution of single cell copy numbers than amplifications that were driven by chromosomal amplifications. And so we thought we could use this kind of principle to make inferences as to whether or not amplifications on ECNA or on the chromosome. And just showing proof of concept, this is our Colo 320 DM cell line versus the isogenic pair without the DM, but the amplification on the chromosome. And what we found is that indeed the copy number means were pretty similar to one another, but the variance in the DM cell line was much, much larger. And so we could use this fact to actually infer which cells had ECNA or which genes were on ECNA versus a chromosomal amplification. And so the way we trained a model is, once again, we went to this evolutionary modeling toolkit that we had and generated a large synthetic data set of both ECNA-driven amplifications and chromosomally driven amplifications. And shown right here is just a summary of what this data looks like. I'm just looking at two different parameters of distribution, the mean copy number versus the variance of the copy number. And you can see that these chromosomal amplifications, roughly they follow Poisson distribution. It's just the variance you observe is just the noise of sampling from a count distribution. Versus the ECNA and even mixtures of ECNA and chromosomal amplifications have much larger variances than their mean would suggest under Poisson assumption. And so we treat just a very simple classifier, just a very simple multi-layer perceptron on neural net to make predictions whether or not an amplicon or a gene seem to be following an ECNA distribution versus chromosomal distribution. And this works really quite well compared to just simple baselines and overall in our simulations. And so we packaged this up into a tool that we called SCAN, single cell amplicon. Now, the first set of experiments was going beyond just the synthetic data set that we generated. Could we start predicting on cell lines that we are actually sure of what genes are on ECNA versus not? And so this is this Colo 320 DM cell line. All these genes in red here, MYC and PB21 included, we know that these are on ECNA, and our model accurately predicts that these are on ECNA. We can look at a panel of cell lines that we profiled with single cell attack sequencing. some with this DM, some with this HSR variant, which means that the gene is on the chromosome, and some other types of tumor types, including glioblastomas, gastric cancers, breast cancers, and neuroblastomas. And we found, by and large, our SCAMP toolkit actually agreed very well with prior characterization, including both from the genome sequencing as well as fish assays as well. With one exception, this BT474, it's breast cancer cell, and that carries an ERBB2 or HER2 amplifications. We did not have any metaphase spread information on this data on the cell, and all we had was whole genome sequencing that seemed to suggest that this ERBB2, shown right here, this is the breakpoint graph, this ERBB2 was carried on ecDNA. And this is a very complex breakpoint graph. If you haven't looked at the breakpoint graphs, I guess you have to take my word. It's a very complex cycle. I don't think anyone can just look at this and say that it looks like ecDNA versus some other complex rearrangement, but our algorithm suggests that this was ecDNA. But if you look at the copy number distribution, it was very clear that this ERBB2 amplification had very high copy number mean, but the variance was really quite low from cell to cell. And so we actually went in and did metaphase spreads looking at the context in which this ERBB2 is amplified, and we found unambiguously that all the ERBB2 amplifications had co-localized with chromosomes and had actually integrated into the chromosome. And so this actually seems to suggest that our single cell assay, not only is it going to open up new questions, but it also offers higher specificity at classifying ecDNA than whole genome sequencing based approaches. And there's some nuances here, I think, that are also quite interesting. I'm happy to talk about later. Okay, but what kind of biology can we start to do with this type of technique? I'd like to walk you through our analysis of about 73 patients that were profiled with single cell TAP sequencing from TCGA, and what we can learn about how ecDNA are affecting the patient tumors. And there's three major questions I'm going to tell you. The first one is how are these ecDNA distribute within a tumor? How frequent are ecDNA in the tumor? The evolutionary history of these ecDNA and the molecular consequences. And so shown right here is kind of a summary of the cohort. And shown in red is our prediction of how frequently we see ecDNA amplified tumor for a particular tumor type. And gray is what's coming from whole genome sequencing. You see that they by and large agree with one another. There's some notable discrepancies, and I have some thoughts as to why that's happening. But even just assuming that that whole genome sequencing was operating perfectly, this single cell assay is going to give us new insights. And the first one is the frequency of how often ecDNA appear within a tumor. Shown right here is about 14 different tumors that appear to have ecDNA given our single cell analysis. And you can see that there's a good fraction of tumors that have very high levels of ecDNA within the tumor. Almost every single cancer cell has this ecDNA. And surprisingly, we also saw some examples, like this LUAD sample, lung adenocarcinoma sample, that had very few cells with ecDNA. And I think perhaps the most interesting example are these that have two different types of ecDNA distributions. Some ecDNA that appear in about 80% of cancer cells, and then some that only appear in about 10%, which is often below the threshold of detection from whole genome sequencing. So we can dig into that one that had these two different pools of ecDNA, and what I've done is just kind of a very simple evolutionary analysis by clustering the copy number matrix. And what we find is actually there's 4 distinct clusters that emerge that each have distinct copy number changes, ecDNA changes specifically. In this first cluster, we can see it's mostly early stage cancer cells that I think don't have any real indication of strong copy number changes. You see that there's some fraction of cells that start to generate some copy number changes, but by and large, these are cells that don't have large copy number changes yet. But you can see we have a second cluster that seemed to have a region on chromosome 12 that may amplify ecDNA that largely carries MDM2. It's a negative regulator of p53. We see another cluster of cells that carry this MDM2 amplification and a MYC amplification. So it's gained another amplification from chromosome 8. And this is a separate ecDNA. So these aren't merged together, it's completely separate ecDNA. And then lastly, we see another cluster here that has this MDM2 amplification region. And then it seems to have another region on chromosome 12 that carries CDK4, which is a cell cycle control. And then what's interesting about this is this implies an evolutionary history as to how these tumors developed with ecDNA. One could postulate that these tumors began with an MDM2 amplification that maybe started or appeared to kind of sweep the population. And then subclonally, there was 2 other amplifications that appeared. One that carried a MYC amplicon and another one that carried CDK4. And that these distinct pools of cells are interacting with one another in the existing tumor that we actually observed. And we can start to postulate how selection might be actually shaping the future of these tumors as well. But I can say that another interesting thing is if you look at the UMAP of chromatin accessibility, what we can see is that the cluster 3 that carries MDM2 and MYC appears to cluster separately from not only the cells that are early stage without ecDNA, but also from the rest of the cells that only carry MDM2 amplifications, or this MDM2 and CDK4. Which really does seem to suggest that the presence of ecDNA is actually controlling the molecular fingerprint of these individual cells. And so we've done some analyses as to how these cells with ecDNA are looking molecularly different from cells without ecDNA. Once again, this is actually really driven by the fact that we have single cell resolution and we can segregate cells with or without ecDNA. And so we can take this mixed tumor population that consists of cancer cells with or without ecDNA, and we could in silico separate those cells with ecDNA versus those that don't. And we can do a differential analysis as to what gene programs are activated. And by and large, this is just a hallmark set. There's a lot of signatures that are not really meaningfully changed between ecDNA and non-ecDNA cancer cells. But there's a cluster down here that seems to be actually really highly expressed compared to cells without ecDNA. And shown in here, there's a couple of interesting gene sets. Glycolysis, which upregulation of glycolysis, glycolytic pathways would seem to suggest that differential metabolic activities in ecDNA cancer cells, as well as enrichment of hypoxia signaling and other signaling that seems to suggest that these cancer cells are growing faster. Now, this, I just as kind of an aside, as we're kind of getting to the end of this data here, I just want to say that we can also apply this to any kind of attack-seq assay. And so shown right here is actually a spatial attack sequencing assay where we can project ecDNA copy numbers onto an actual GBM sample and look at the spatial niches that ecDNA appear in. And this actually does seem to suggest, if we look at VEGF-A, which is a marker of hypoxia signaling, that indeed that ecDNA cancer cells are existing within these hypoxic niches. And so between these two pieces of data, between the increased chromatin accessibility of hypoxia gene regulons, as well as the spatial context, really does seem to suggest that these ecDNA cancer cells are adapting to their microenvironments and responding to environmental cues. Perhaps even these environmental cues are creating or actually causing new ecDNA to emerge. And so as I'm kind of closing here, I just want to say that beyond molecularly profiling ecDNA and cancer cells, we also think that this is broadly clinically useful, this copy number distribution that we can get from cancer cells and predicting whether or not ecDNA or chromosomal amplifications are driving the copy number distributions. And so kind of contextualize this a little bit, it's actually been a major challenge to identify which cells have ecDNA or which tumors have ecDNA versus which tumors do not have ecDNA. And as we're getting to an age where we're starting to be able to target tumors with ecDNA, this is a major clinical challenge in order to stratify patients that actually might be good candidates for these ecDNA target pharmacological targeting agents. And so this is really also difficult because it's really a challenge to do metaphase spread FISH assays from clinical samples because that requires actually culturing the cells, resting in the metaphase, and oftentimes we're not culturing tumors and doing these assays. And so we thought perhaps we could get good enough copy number information from regular DNA FISH, which is a routine assay in clinical applications, and maybe we could predict which tumors actually have ecDNA versus which tumors don't have ecDNA. So we took a very simple tumor microarray, about 12 patients across a variety of different cancers, some that had indicated that they were amplified for MYC specifically, either by sequencing with panel sequencing or by DNA FISH. But of course, this DNA FISH doesn't tell us if it's ecDNA or chromosomal amplification, it just tells us if the signal's higher. So we took our SCAMP model and tried to predict from these copy number distributions if there was an ecDNA amplification. And what we're really struck to find is that we could actually partition these tumors into those that appear to be driven by ecDNA versus those that did not seem to arise from ecDNA. And I should say that when we look at the actual FISH signal, we can see that this 108 sample right here that has a very clear signal of looking from ecDNA really does seem to have several distinct foci of MYC, which seem to suggest that this is being driven by an ecDNA amplification. We've also looked inside of xenograft tumors as well that were driven by our estrogenic cancer cell lines, and that also works as well. So we think this is actually goes beyond just molecular classification of cancer cells with ECA, but also could actually be the foundation for new clinical assays for diagnosing tumors as either ECA-driven or chromosomal-driven. And so, yeah, we think that STAMP is really a flexible toolkit. It has clinical application, but also experimental applications. Allows us to do quantification of ECA-positive cancer cells, allows us to look at the clonal dynamics within a tumor, when ECA emerge or when they die off, and it allows us to do phenotyping, both single cell and spatial phenotyping of cells with ECA. And we think it's, at least for the immediate future, as we're using these types of approaches to look at ECA, we think that this kind of toolkit, as we continue to build it out, will really allow us to bridge these two different visions we have to merge both space and time measurements with predictive models. And so just in closing, and I'm happy to take questions, I just want to acknowledge everyone who's been involved, especially King Hung and Natasha Weiser. King was an excellent graduate student, now a postdoc at Scripps, who co-led the co-segregation study with me along with Ivy, as well as Natasha has been an excellent clinical collaborator who's led a lot of work with STAMP with me as well. And then Katerina and Sedona were just excellent colleagues as well, who led this ORCA reconstruction of and discovering of transposal elements in ECA cancer cells. I'd be remiss not to also acknowledge just the excellent advisors that I had during my postdoc, Paul Michel and Howard Chang. I'd like to thank my slowly growing group of excellent graduate students and undergraduates, as well as I acknowledge my disclosures at Tahoe and Vector Biotechnologies. And happy to take any questions should you have them. Thanks so much. Please, Jose.
Yes, so I have a question about your but regarding the evolutionary, one that you thought about speaking in terms of rational design therapeutics for subclinical. Seemed like that the example you gave, right? It seemed like, well, you must have something to CDK4 inhibitor.
Yeah.
And I'll just roll on the second question.
Sure.
So it's rare, well characterized progressive figures. What about premium plastic? Like, is there some prognostic, values that you see it?
Yeah, phenomenal question. So the first one is that I think the dream is to be able to do rational design of therapeutic strategies based off of the ECA content. And currently, maybe STAMP is the method we use, maybe there's another method, but there's no real way of being able to profile the composition, the subclonal composition of ECA within a tumor. And so that in itself is a challenge. But then I think the mandate is to also do experiments and study how different combinations of ECA within a cancer cell or within a tumor are going to respond to therapies, but also microenvironmental pressures. And so one of the things we're really excited to do is start leveraging new technology that allows us to write ECA within cancer cells. Two of these include CRISPR-C, which is an approach that uses Cas9-induced double-stranded breaks to launch regions within the genome. And there's another set of techniques that have been pioneered here by using Crea combinase to launch these segments as well. And so we have new molecular techniques that allow us to launch ECA and then tying these with pharmacological agents and kind of microenvironmental exposures, I think, are a major opportunity to start doing the rational discovery. The second thing is about, do we ever see ECA in preneoplasia or tumors that may be on the verge of becoming real full-blown hyperplasia? And the answer is yes, but it's also comes down to detection as well. So the example that we often give is in the case of Barrett's esophagus. There was a study, I think, published in 2023, I think it was, that showed that actually the transition from Barrett's esophagus to esophageal cancer was really mediated by ecDNA amplification, which is really quite surprising because for many years we thought that ECA was maybe an event that happened later in tumor development, once a lot of the gardens of the genome have been mutated, or this cancer cell is able to accommodate several different types of kind of genomic insults. And so that's really just to suggest that ECA can emerge early on in cancer. But if that happens, it might be at a low copy number, and it might be difficult to detect from whole genome sequencing as well, because not only low copy number, maybe just one or two cells. And so I think this is another example of something that single cell or has spatial resolution will really allow us to answer that question: is what role does ECA have in the pre-neoplasia context? Yeah. Thank you. Yeah, please.
Sorry, this is probably a more fundamental question about EPDNA principle. What controls like either the formation of like the excision of the genome form these things? And you mentioned about like reintegration upon the treatment.
Yeah.
Like it's not like the cell is just like, oh, I want to be safe, so I'm going to reintegrate this. There is some random stochastic process that's possibly happening, or is there like just a baseline? Like there are like a small population of cells that have FGFR in the genome right from the get-go, and that population is the one that's coming up.
Yeah, so phenomenal questions as well. So our understanding of ECA emergence is a bit opaque, but there's several mechanisms that have been described in the literature. I think two major mechanisms are worth mentioning here. The first is chromothripsis, which is well documented and certainly can generate ECA. It might be one of the major drivers as well. And so basically how people think this happens is chromothripsis is a, the shattering of the genome, catastrophic shattering of the genome. And the thought is that lacking chromosomes during actual cells segregating can be encapsulated into these micronuclei structures that exist in the cytoplasm, cytosol, during interphase. And then you could have several different DNA fragments that can encapsulate in that if you had several lagging chromosomes. And then there's processes by which the micronuclei envelope will rupture, and DNA damaging agents will enter to the micronucleus and induce double-stranded breaks or DNA damage, but also ligation of these fragments together. And then at some point in a future cell division, these kind of resulting restitching of fragments will re-enter into the nucleus and either integrate back into the genome or exist as ecDNA. So chromothripsis is one way. The second way is you could just have random double-stranded breaks in the genome that could induce ecDNA. And this could happen in the context of chemotherapies, let's say, that induce DNA damage. It could happen just from other kind of environmental exposures as well. But this is kind of what we call the excisional pathway of ecDNA biogenesis. But our understanding of the different rates, the context which within these pathways are favored, is not really well understood. And then your second question is about reintegration. That's also not very well understood. What I can tell you is that I was a big disbeliever in the data that I showed you until it's important to recognize, and I'll just flip back here. Until we did the experiment where we actually created these monoclones right here. So we took the parental cell line and we created monoclones where they only had ecDNA amplifications. There was no way that there was residual FGFR2 amplification on the chromosomes. And so the question is, like, how often does this happen that you have integration? People have identified this pathway before, and I think that it's largely due to DNA damage that occurs, that there's double-stranded breaks in the chromosome that might introduce the opportunity for this ecDNA to integrate, but we don't know under what contexts are actually favored or how often this happens, and in what genomic context they're more likely to happen versus others. So there's another active area of research. And as to why they're favored in drug conditions is also a largely unknown. Great question. Please, go ahead.
Sorry if I missed it when you talked about the idea of can you see that after you start and then you still can use that the observation as well? Do you elaborate a little bit more on that and why that happen?
Yeah, so I think that the thought is that ecDNA are kind of a bet hedging strategy for cancer cells. So you have very variable copy number distributions, and allows the cancer population to kind of change their strategy for what copy number like mode to select for, depending on their environment or other types of things. And so almost ecDNA cancer cells exist when the environment is most unpredictable. So I always joke, like, I should be the one culturing these ecDNA cancer cells if you want them to be around, because who knows what I'm doing in the hood and stuff like that. And no one took me up on that offer. I'm kind of glad they did it too. But so basically, in the context of the drug holiday, what we're doing is we're changing the environment in between treating the cancer cells. We take off, take off. And so in that sense, the cancer cells can't really commit to a stable strategy. And so that's why I think we still see the ecDNA around, is because we're in effect selecting for cells that have maintained the bed hedging strategy. And so in one way, it almost is like the drug holiday is, it almost sounds like what we're doing is trying to keep ecDNA-driven cancer around. But in reality, I think my interpretation is it means that we're able to just control the population better, because it's once the cells have integrated the ecDNA chromosome that we lose ability to target the cells at all. So it's a trade-off between keeping these ecDNA cancer cells around versus being able to control them. Yeah. So hopefully that answers. That's my interpretation of data. Is there another question over here? Yeah, please.
And they had similar questions, but how can you tell if the ecDNA is driving the tumor growth, or maybe any particular step of tumor growth?
Yeah.
And is there a kind of a threshold copy number that's required for that?
So, I mean, phenomenal questions. I mean, I think that it's totally reasonable to wonder, how much of the phenotype we're seeing is actually due to ecDNA itself versus it's just a copy number effect. And one of the things I'll say is that I think both can be true. Is maybe copy number is largely driving, it could be largely driving the phenotype that we're seeing. But ecDNA have a remarkable ability to remodel our copy number distributions to the optimal state. And so one example of this is that if you grow these colorectal cancer cell lines in vitro, the DM and HSR, if you do drug competition, the HSR line often outcompetes the DM line because every single cell is optimized around that in vitro condition. But then if you put these cells in vivo into a xenograft tumor, it flips. The DM copy numbers, or the DM ecDNA cancer line, is able to take over. And so I think one of the ways I can answer that question is saying that the copy number distribution is much easier to toggle and titrate in the ecDNA cancer cell line or cancer context. And so beyond that, though, there's also the other data that I think is pretty compelling that seems to suggest that ecDNA themselves are able to cultivate a really permissive epigenetic states that drives increased gene expression per copy number of whatever molecule it is. So to me, it's these two. Each copy is itself more productive, as well as the ability to actively titrate the copy numbers. Is that answer your question, or do you have a?
Yeah, I think so. I mean, I guess the question is, can you, like you had PBT on those circles, how can you tell that PBT, this may be an extended question, but how can you tell that PBT is required for the tumor growth versus the NIC amplification?
I see. So what's actually essential on these circles, maybe? Versus, and I guess MYC is an obvious one. MYC is a known oncogene, but if we see circles that carry other kind of amplifications, like how do we know if it's functional or not? Yeah, and I think that's a great question. I think that some of the single cell modeling we do, where we can actually tie the distribution to the selection coefficients, is one way of answering that. But it's actually this transposable element story was how I started thinking about that as well. There's tons of junk on these circles. There's these are about 4 megabase in size, and they're just comprised of whatever got excised out of the genome. So you have things that are functional, you have things that are not functional. And a big area, I think, of research is understanding what sequences actually are driving the copy number changes, the function of these ecDNA, the retention of these ecDNA. And we have some indications as to what sequences are advantageous, but I think we're kind of at the start of that as well. Big opportunity. Thanks. Go ahead, Dan.
If we talk, I find it's fascinating to talk about this, like the post-aggregations. So one question I have is, like, if you target FGFR and you only have MYC there, can two different MYC ecDNA form a hub?
Yeah. So we think yes. We think that we're not treating with triptolide here to break up the hubs. We think the hubbing is still occurring, especially between FGFR2 and MYC, but also between MYC and MYC. And so this is a really insightful point because just measuring the absolute copy number might not actually get to the full phenotype of these cells because they're synergistic combinations. And so that's really quite interesting. I wonder if there is kind of a threshold at which point the hubbing phenotype or the hubbing function actually overcomes disadvantage of having individual copies themselves. There might be phases where hubbing is much more advantageous. Maybe that's also what we're seeing in the drug-resistant standpoint.
Because initially I'm thinking maybe if you lose the adaptive member of the hub, you lose the hub. Maybe that was lead to some changes in the content accessibility or methylation status of the. Maybe you select for some super expressor or some
Totally agree. Yeah, no, and one of the simple things that we're doing right now is doing single cell multiphase after these drug experiments to see if we can see beyond just the copy number dynamics if there's any kind of functional changes molecularly on these intervals. That'd be really exciting. That's the obvious next thing to do. Yeah.
I have one more question, please, online. Very interesting. Oh, multiple questions actually. Super thought-provoking talk. I'm curious if ecDNA has any higher propensity to get out of the nucleus and end up in the cytosol.
So that's a phenomenal question and something that has kind of been a big mystery because many of you might probably appreciate is that our cells have tons of cytosol-like nucleic acid sensing pathways, including cGAS-STING, which should detect cytosolic DNA. So you have tons of ecDNA. And they're all, I didn't say this, but they're actually organized around the nucleus and like the perinuclear or they kind of around perinuclear, so like on the envelope. So they're in a position to kind of leak into the cytosol. Why don't we see more of the cGAS-STING pathway active? So it's a major mystery. We're looking at that. It seems like, though, that some of the modeling we've done seem to suggest that even just small deviations where ecDNA can leak into the cytosol would lead to depletion of the copy number over time. So something in the cancer cell is allowing these ecDNA to be retained faithfully over time. So it seems like they might have an increased likelihood compared to chromosomes, but for some reason they're not really leaking into the cytosol all that often. Yeah.
Okay. Second question, very interesting topic. Is there an indication for how ecDNAs might replicate together potentially, and are they line or other non-coding element dependent? Can your single-cell ATAC-seq combined with single-cell RNA-seq find gene expression differences to uncover necessary genes/proteins for ecDNA replication?
Phenomenal question. So these are actually like the questions that we have students looking at right now is well, what's the landscape of repetitive element those elements on ecDNA? What if they're actually enriched across cohorts, they have functional bearing, and then also trying to figure out what are the molecular factors that are important for the retention of ecDNA across cell divisions. Now, ecDNA do replicate, and that's very well known. We think that they replicate just like regular chromosomal DNA. They replicate during S phase. They might be, largely kind of late replicating regions. They associate with laminar associated domains, and so they might be late replicating during S phase, but they're still replicating during S phase. But it's really quite interesting that they are retained over time and replicated exactly the way of chromosomal DNA. So these are questions that we're looking at right now. Phenomenal question.
Thank you.
Thanks.
Yeah,
Okay, great. Thanks so much all for your